The final products of most genes within a cell are proteins, and the cell's function is dictated by its collection of proteins. Proteins are composed of amino acids, and a protein's function is dictated not only by the sequence and composition of these amino acids, but also by the proper three-dimensional structure formed when these fold back upon each other. Disulfide bonds are bonds that occur between the thiols of cysteine residues of a protein, and these play an important role in both the stability of a protein as well as protein folding. I'm doing my research on PDI, or protein disulfide isomerase, which is able to catalyze the oxidation, reduction, and isomerization of the disulfide bonds of proteins. PDI is a member of the thyroidoxin superfamily, and it is localized to the endoplasmic reticulum, the plasma membrane, as well as the extracellular space. It is composed of 5 domains, and 2 of these domains, the A and the A' domain, have catalytic activity through a CXXC motif. The exact mechanism of this catalysis is unknown. However, previous research in Dr. Mutis's lab has shown that there are 2 critical lysine residues, one which flanks the A domain CXXC motif, another which flanks the A' domain CXXC motif, which are required for PDI's optimal catalytic activity. I'm going to be studying the effects of lysine modifications on the activity of PDI, and in order to do this, I'm going to utilize site-directed mutagenesis as well as chemical acetylation by treatment with acetic anhydride in order to mutate PDI. These PDI mutants will then be compared to wild-type PDI in both chaperone and kinetic assays in order to determine how the mutations affect PDI's activity. This research provides insight into potentially critical lysine residues which may regulate PDI's oxoreductase catalytic activity through interactions with the CXXC motif. The final products of most genes within a cell are proteins, and a cell's function is dictated by its collection of proteins. Proteins are composed of amino acids, and a protein's function is determined not only by the composition and the sequence of these amino acids, but also upon the final three-dimensional structure that forms when these fold back upon each other. Disulfide bonds are bonds that occur between the thiols of cysteine residues on proteins, and these play a very important role in both the stability and the folding of a protein.